So, hello everyone. In the last lecture, we are discussing structured kinetic models. So, if you remember, in the case of unstructured one, it does not give information about dynamics of the cell biomass. Rather, it considers cell as a black box model. But if you want to study or get more insights, then we need to discuss structured kinetic models, but there are this n number of metabolic reactions, then how to segregate them. So, one option we discussed in the last lecture was based on the relaxation time. So, we can divide genetic system, epigenetic system and metabolic system based on the order of time scale. So, highest time scale is for the genetic system and lowest is for the metabolic systems. Further we discuss, based on this relaxation time, we can divide into different groups, and typically it is found that 2 or 3 variables are sufficient to model a system, and based on that, in the last lecture we discussed 2 compartment models. The first one was the Williams 2 compartment model, and second one was Ramakrishna. So, in the Williams compartment, the first compartment included RNA and second compartment included DNA and proteins. While in the case of Williams model, what we discussed, the first compartment included RNA and DNA, while the second compartment included proteins along with inhibitors. Now, today we are going to discuss how to do the structure modeling based on the metabolism, and then finally we will compare unstructured and structured models. So, first we are going to discuss models of cellular energetics and metabolism. So, as you know, based on the metabolism, one option is to distinguish like respiratory and fermentative processes. So, before going ahead, we will be discussing very brief about the metabolism. So, metabolism is a biochemical pathways and through which the cells harvest and invest energy through 2 complementary networks. First one is the catabolism and second one is anabolism. Catabolism means breakdown. So, here a substrate is converted to carbon dioxide, other byproducts as well as this energy generating molecules like ATP, NADH, NADPH are produced. Few example of substrate includes carbohydrates, hydrocarbon and other reduced carbon containing compound, let us say glycerol. Anabolism, in this case the precursors are self assembled to synthesize biomacromolecule or biosynthesis process, but here it requires the energy and energy is supplied through ATP. So, different biomacromolecules like DNA, RNA, nucleic acid, these are nucleic acids, lipids, carbohydrates and different intermediates. So, basically what we are seeing is ATP is produced during the catabolism and it utilized during the anabolism process. So, we need to see the carbon flux, energy flux and redox balance. One example is, let us say, simply glycolysis followed by this TCA cycle and leading to the synthesis of biomass. So, what it says, this energy rich compounds serve as a link between breakdown and biosynthesis, and here we discuss the energy rich compound ATP, GTP, NADH, NADPH. So, let us take example of catabolic process. For example, in the glycolysis, glucose is converted to pyruvate, it yields 2 ATP and 2 NADH molecules. In the TCA cycle, oxidizes acetyl CoA to carbon dioxide and generates ATP, NADH, FADH2. And further, if it is going through oxidative phosphorylation, then we see this how this NADH and FADH2 are further converted to ATP, and it gives to the large amount of ATP flux. So, it says the regulatory control ensures a balance between energy supply and energy demand. So, for example, if you see during the glycolysis only 2 ATP is produced, but if we club glycolysis along with oxidative phosphorylation, so it says the regulatory control ensure a balance between energy supply and demand. For example, if there is a requirement of 2 ATP that can be generated only through glycolysis, but once the glycolysis is combined with oxidative phosphorylation, so we get 38 ATP molecule from 1 mole of glucose. So, it says that depending on the requirement, the particular catabolic pathways are selected by an organism. Next is the example of anabolic pathways. We have already discussed this gluconeogenesis and pentose phosphate pathways for the sugar and NADPH supply, synthesis of amino acid from the TCA cycle and glycolytic intermediates, nucleotide synthesis from simple precursors, and they require ATP hydrolysis for the energy supply, lipid assembly from acetyl CoA and glycerol intermediates. So, these are the examples of anabolic pathways, and all these pathways require energy supply that comes from this energy settling molecule, that is ATP. So, flux through these routes depend on the precursor availability as well as the energy status. So, what it says, we need to integrate this coupling energy and biosynthesis. So, both anabolism and catabolism need to be integrated together. Now, when we are going to model based on this cellular metabolism, so what it says, you can divide this metabolic process in terms of either respiration or fermentation. So, it says the level of metabolic detail required increases beyond the compartmental models. What we discuss in the compartmental models, this synthetic component or synthesis component. Further, it will be useful to consider the type of reaction that occurs within the cell, and metabolic pathway, as we have discussed, can be distinguished as catabolic and anabolic, and this energy producing and energy consuming processes must be tightly regulated within the cell. Further, it is necessary to consider both carbon and energy flow within the cell when we are developing these complex structured models. Now, what it is going to do? It is going to have this compartmental framework group metabolites by function or location. So, here it is going to group based on the function, and then we have to see dynamic balance for the both mass and energy flow across the reaction, and third is these regulatory steps that is represented by the kinetic terms, rate constants. So, what is the advantage? It predicts this unbalanced growth phases, and in addition it simulates the transient response against environmental changes. So, the one model based on metabolism is aerobic growth of the yeast Saccharomyces cerevisiae. So, if we just see this one, if mu is less than or equals to the mu critical, then this becomes a fully respiratory process. In fact, RQ is very close to 1, Yx by S is 0.5. So, it means say the low growth rate, and what is the product of this process is the biomass and carbon dioxide. But at the same time, if you just see if mu is greater than the mu critical, so we are going to see the mixed process, and particularly it is dominated by the fermentative synthesis of ethanol, and in this case what will be the product will be ethanol and carbon dioxide. So, it says for the yeast, at least based on the function, we can say it is either respiratory, either it is a respiratory or it is a fermentative process. And how it is happening? It is happening due to change in the enzyme pattern. This switches from respiration to the fermentation. Now, typically if you see the respiratory enzymes such as isocitrate lyase, malate dehydrogenase and cytochromes are repressed at high growth rate, and here the glycolysis provides the main source of energy. Now, at the low growth rate we see the reduced level of glycolytic enzymes. So, one option is how it shifts from respiratory to fermentation process can be explained by this Crabtree effect. So, earlier what we discussed, the Crabtree effect was when there is a high concentration of glucose that causes the substrate inhibition, but it is also said that because of this enzyme repression at a high catabolic flux, that is also causing the Crabtree effect. So, Crabtree effect was initially thought to result from the glucose acting as catabolic repressor, but now what we are seeing is a high catabolic flux is directly causing the respiratory inhibition. Glucose concentration play a secondary role. So, it says both this high catabolic flux plus high glucose concentration both are responsible for this Crabtree effect. Now, how we are going to model here? The model is based on two-stage breakdown of cell cycle. The first one is G1 phase. This depends on the availability of limiting substrate, and second phase is assumed to be independent of substrate, and it includes the length of division phase, which is sum of G2 M S phases. Now, what are the two compartments here? We are saying A mass and B mass. A mass carries out the substrate uptake and energy production, and B mass carries out reproduction and division. So, we can say this A mass is a catabolic and energy functions, and B mass is having reproductive functions. Further, B mass is converted to A mass at a constant rate, whereas A mass consumes the substrate and produces B mass at a variable rate. So, based on this, we can write this A mass is converted to the B mass, and it utilizes the substrate A1 fraction of substrate to B is particularly it is a biomass. Then further this A mass is again converted to B mass, but what is difference here? Here there is no substrate. So, once the substrate is depleted, the E is reflecting the ethanol is now being utilized as a substrate, and it is converted to B. And third is the parallelly B is separately converted to A. So, A plus B is the total cell mass X, and E is the ethanol concentration. So, you just say the cell is comprised of two compartments A and B. Now, in the respiration, just see what is happening. A plus, let us say example glucose, oxygen, it is converted to B, carbon dioxide and higher ATP yield. And in case of fermentation process, let us say A is reacting with glucose in the absence of oxygen, producing biomass along with this ethanol, that is your product, and lower yield of ATP. So, you just see the first expression reaction is this is a fermentation, and the second one is this is a respiration. And of course, these two reactions are regulated by the substrate flux as well as the enzyme capacities. Now, based on these equations equation 1, 2 and 3, the following rate expressions are assumed. RA is, we are assuming it is following the monod type kinetics. RA is K1 A S upon Ks plus S. RB is K1 A, the substrate is replaced by the E, this is Ke plus S, and RC is K into B, because B is converted to A in the third reaction. So, the budding process B to A is assumed to occur at a constant rate, where respiration and fermentation follow the monod type kinetics. Now, we can write the mass balance for each of the species assuming a constant cell volume. So, now we can write dB upon dt, dA upon dt, dE upon dt, ds upon dt. So, dB upon dt is twice of Ra plus twice of Rb minus in Rc B is converted to A. And if I see in terms of, if just see B, there is a 2B here, 2B here. So, 2B here, 2B here, and 1B is going this converted to A. So, it says twice of Ra plus twice of Rb minus Rc. While in what can be in the case of A is produced in the Rc, but in Ra and Rb it is depleted. So, minus sign is there. Similarly, A3, similarly E, DE. E, here E is produced in the case of Ra, and here E is utilized, and what is the fraction of A2 is produced in the reaction 1 and A3 is utilized in reaction 2. So, this will be A3 into Ra minus A4 into RB. So, this will be A3 into RA minus, rather it will be A2 into, this will be A2 into RA minus A3 into RB. So, you just see here A2 is produced here and A3 is consumed here. And for ds upon dt, it is only in the reaction 1. So, this will be in the minus because it is utilized. So, it will be minus A1 into RA. So, these are the 4 rate equations, and if you can solve, then you should be able to predict the profile of A, B, E and S. So, here from these expressions, what will be the yield of the fermentation process? The first one, it is 1 upon A1, the substrate, which is fraction of stoichiometric coefficient of substrate, that is A1, and the value of A1 from the experiment was found to be 6.67. So, this is 1 upon 6.67. Similarly, the Yx by S in the respiration, this will be the stoichiometric coefficient of substrate, that was the ethanol here. So, this is 1 upon A3 and A3 value was reported to be 2.22 and A2 value was found to be 2.80. Further, one can calculate the specific uptake and production rate. The Qs is defined as 1 upon x upon ds upon dt. What is ds? ds was minus A1 RA divided by x. Similarly, one can calculate the specific uptake rate of ethanol. It will be 1 upon x upon dE upon dt, and dE is A2 RA minus A3 RB upon x, and specific growth rate can be calculated mu as 1 upon x dx upon dt. In addition, this model enables us to calculate specific oxygen uptake rate and specific carbon dioxide production rate. So, specific oxygen uptake rate, oxygen uptake is taken, oxygen uptake was part of equation 2 where the rate is RB, let us say fraction is alpha upon x. And what is alpha here? Let us say this, if you want to see how many moles of carbon dioxide, let us see the reaction 2 here, yes. Then what will be the alpha here? In the respiration, complete oxygen of ethanol to carbon dioxide and water requires C2H5OH. This is converted to CO2 plus H2O. It requires 3 moles of oxygen. How many moles of carbon dioxide will be produced? 2. This will be 3H2O. Okay, all right. So, it says now from here we can calculate the value of alpha. Alpha will be 3 upon 2 equals 1.5. So, it says the value of alpha is coming out to be this 1.53 here. So, in this way we can calculate specific oxygen uptake rate, and specific carbon dioxide production rate can also be calculated, because here we have to do the carbon balance, carbon in carbon dioxide, carbon in glucose, carbon in ethanol and carbon in cells. So, if you just see Qs, you can calculate in the, what is the fraction of carbon in glucose is 0.4, fraction of carbon in ethanol is 0.52 and fraction of carbon in the cell is 0.48 divided by 12. So, in this way this model also enables to calculate specific oxygen uptake rate and specific carbon dioxide production rate. So, you just see this is the model, predicted model parameters. This is a biomass X, this is the substrate and this is the ethanol. So, it says initial glucose concentration was close to 20 grams per liter, ethanol was 0 and dry biomass was close to 0.15 gram per liter. Now, you just see in the lag phase there is a minimal biomass increase, very slow increase here up to 0 to 2 hours, but when we are seeing the exponential phase we are able to see 2 to 8 hours their rapid growth and rapid fast intake of glucose. And now glucose during this phase glucose concentration has significantly decreased, let us say from 20 to 5 gram per liter, and ethanol remains nearly 0 because this is a respiration phase. So, you just see during this phase in the respiration there is no ethanol synthesis. Next, what we are seeing in the 8 to 10 hours, this is a diaxic shift we are seeing here. We are seeing a diaxic shift here, and glucose is nearly exhausted and ethanol now begins to accumulate here. Now, we are going to see further secondary growth here. Now, how this secondary growth is taking place? Now, this secondary growth is taking place because sugar is already exhausted. Now, ethanol consumptions. So, there is a consumption of ethanol is taking place. So, what it says, this model is this very well able to predict this diaxic shift particularly and more detailed dynamics of system, particularly this lag phase, exponential phase, diaxic growth and stationary phase. And when you just see ethanol is 0, cell mass has reached to stationary phase. In addition, as we discussed, it also gives the information of RQ and the specific gas exchange rate. This is RQ, this is QCO2 and QO2. So, what we are seeing is this QO2, it rises steadily during the exponential phase, then further it peaks just before glucose depletion and further it drops as the respiration stops and fermentation begins. It increases, then further it stops here. Similarly, if you just see this CO2, it simply follows the reverse of QO2. You can say mirrors of QO2 in the early phase. It was increasing and it was decreasing, and it exceeded to the QO2 value during the fermentation phase here, and it exceeded here. That's it. Similarly, RQ, if you just see the RQ, it maintains unity during the fully oxidative process and it climbs to 1.5 during fermentative overflow and again falls back towards 1 during the ethanol reoxidation. So, it is like here and further it goes here. So, what it says, we are able to see the profile of this, predict the profile of QO2, QCO2 and RQ value as well. So, this was the example in the batch culture. Now, continuous culture was also modeled. So, in case of continuous there are few differences, particularly the K2 value and A3 value. So, K2 value here it was in the batch 0.3, it has become to 0.5 hour inverse, and A3 value was 2.22 in the batch and it become 1.65 in continuous culture. So, here you just see the x axis now change, here is dilution rate and y axis is the biomass and specific gas exchange rate, and further you can see the profile of ethanol as well as the glucose here. So, we will quickly explain these figures. You just see the biomass profile of X, it increases to a maximum value near D equals to 0.25, then declines towards, it reaches maximum here, then further it declines towards the washout. Glucose, it is high at low value of D, you just see the glucose, it is high at the low value of D, and then further it depleted very sharply. Reverse profile will be for ethanol, it is 0 at initial dilution rate, then it climbs during the fermentation metabolism, it is here, and further you can see there is a slight drop here. Similarly, you can see the QO2 and QCO2, they follow the reverse profile. QO2 increases to saturation and QCO2 rises faster once fermentation begins. So, you just see the profile of, this is the QO2 and this is the profile of QCO2. So, here during the fermentation process QCO2 is rising and QO2 is declining, and the RQ value is 1 at the low value of D, it jump beyond 1 at the critical D where ethanol forms. Now, what is the critical, what are the key insights here? The first is the critical dilution rate. So, it marks the onset of shift between the either respiration or the fermentation process, and biomass yield drops and it is converted to ethanol during the fermentation process. So, in the respiration biomass is produced, in the fermentation ethanol is produced, and model captures both the threshold behavior and quantitative trends of gas exchange as well as the product formations. Now, we can compare all three models. So, in case of unstructured monod model, equation we already know, mu is mu max S upon Ks plus S. So, it assumes cells as a black box, but it also consider the rate limiting substrate, and if we just see the profile of monod, it predicts the exponential phase followed by the plateau phase when substrate concentration increases further. Logistics we have discussed earlier, it says particularly about the X is reaching to the X max following a exponential profile, but it is independent of substrate. There is no explicit substrate or byproduct terms here. And now, in the case of structured two compartment model, particularly this based on the metabolism, it splits biomass into A catabolic function and B the reproduction, and we assumed two parallel pathways, respiration as well as the fermentation, and then we did the mass balance for the A, B, S and E. In addition, the gas exchange rates, and followed for the respiration and fermentation we assume the monod type kinetics for each route. So, if you compare here all three models, particularly you just see based on the substrate, it is independent and these two are dependent on the substrate, and structured model is able to predict the biomass formation while these two models cannot predict. And what are the different phases? You just see structured model is able to predict lag phase, oxidative, fermentative and second growth phase. But this complexity increases as we go for the simple to the complex model. I just see the number of parameter was 2 in logistics, it became 3 in monod, and in the structured model it becomes more than 8 plus. But it is important if you want interested in predicting metabolic profile, gas fluxes and yields, one should explore this structured growth kinetic model. So, what it says, this logistic model is a quick, you can easily do the curve fitting, but it does not give any biochemical insight. Monad, it incorporate the effect of limiting substrate, and it is very good for single substrate chemostat studies. And this structured two component model, it is necessary when byproducts and metabolism shift matter, and it captures interestingly diaxic growth, gas exchange as well as the yields. Further, this unstructured model is a simple, and this structured model becomes this more complex. So, here you require fewer experiment, here you require this more number of this require extensive kinetic and stoichiometry data, and further this as the number of unknowns are more here, you require the high computational and experimental data. So, with this I think I compared this structured and unstructured model, and depending on the experimental data and what insights are required, one can choose to apply either unstructured or structured kinetic models. Thank you.